Yes.
So
I don't think
I basically work with human genetics. I
Mhm.
work with care genes and HLA.
Mhm.
And in my PhD, I used to work with population genetics.
Mm.
But now that I'm a professor,
That's
Eduardo,
another working on
Oi.
population genetics.
Sim,
There are,
tá funcionando o compartilhamento.
There are the old professors
Oi?
Antonio Amorim
Tá
in
funcionando o
in Porto. There's
compartilhamento.
another Antonio Amorim in Lisbon, and that's Eduardo Amorim,
tô
who's now in the US also working on population genetics. Yeah.
currently I'm not working with this. But
Obrigada.
I'm planning
Por favor.
to come back to my origins. You know,
Obrigada.
because
Eduardo, vocês estão vendo os slides?
working with population
Sim,
genetics.
estamos vendo.
Em modo de apresentação?
I will tell the
Não,
story
ele
place.
é isso, ele tá no modo de apresentação, isso mesmo.
Tá certinho então, né?
Tá certinho.
E nós já estamos gravando, certo?
Isso, sim.
Ah, então tá bom. Muito obrigada.
MTDNA and forensic genetics.
So you can open your, you know everything that I'm going to tell, so.
2 weeks, your class of the advanced DNA, I'm very
For how long are this subject?
All my scientific life, so that's, so, well, of course, the field has been evolving, but I started at the time in the early 1990s where DNA didn't exist. I worked with HLA, antigens, with blood groups, with protein polymorphisms, and then of course we could move on to a time where that's something like 5 SNPs. That was a real doctoral dissertation. Now of course a PhD is something like 500 or 100 genomes.
Yes. Thanks a lot.
Yeah. The types of things you couldn't even imagine in DNA also. And you see a little bit, particularly the first class, the evolution of the two to be.
I think it's gonna be an amazing course. You know?
I hope I don't disappoint everyone.
I'm sure you won't.
Yeah, on occasion, my lectures seem to be wonderful for insomnia.
I used to teach my classes at night, and then I'm speaking, and everyone's so tired that I told people exactly that. My classes are really good for insomnia. Everyone starts sleeping after I start talking.
You think it seems to be working, at least the presentation? It's yeah.
Yes, it will work, but now this is like frozen. Eu acho que eu tenho que desconectar, tá vendo?
Não tá passando o slide com o passador nem com.
Não, tudo deu certo.
Yeah, that's fine. Yeah,
It's working.
yeah, again, yeah, it's.
O
You
compartilhamento
can
parou.
leave me about here, and yeah.
Yeah.
I will watch on the, yeah. So, I will watch on the
Wow.
screen.
Eu
Uau,
acho que esse passador tá com bateria fraca, porque ele.
Tá procurando sticker?
uau.
Eu tenho que falar que empresta o meu.
O compartilhamento parou.
Ah, tem, por favor, Patrícia.
Pode ser.
Porque
compartilhamento.
ele ficou verdinho.
Filho do sonho.
Desculpa, Eduardo, o que você falou?
O compartilhamento parou dos
Ah,
slides.
parou? Tá bom. Eu vou compartilhar.
É o projetor. Esse negócio também. Você esqueceu?
Esse é a professora Angélica.
Tem que trocar pilha, eu acho.
Uhum.
And this is the first time you come through Zoom?
Yes.
Yes. Are you trying it?
Yes, it's went down to several grass and movettes.
Okay,
And
that's good, great.
yeah. And on top of that, we went to this restaurant in Movettes with sort of an outdoor patio. And there became a family of TT monkeys. That was cool.
Really cool.
Wow.
The nature there is pretty.
Yeah.
And here in Curitiba, you have a lot of parks involved star spaces. So
Yeah. Well, I need to be like to the botanical garden.
yeah, botanical garden is one of the most beautiful parks here in Curitiba.
Mesmo dia, tudo bem? Muito obrigada, Patrícia. Como? Obrigada.
Daí, como tem que colocar online,
Agora acho que foi.
Ok?
Thank you. Eu acho que você está clicado na apresentação mesmo? Está em modo de apresentação?
Tá dando tudo certo ou não?
O passador mas,
Agora funcionou.
mas a gente já trocou de passador.
O micro não tá funcionando?
Então, mas o problema não é. Eu acho que talvez o problema seja a gente mesmo.
Mas ele piscou a bateria mesmo.
Acho que ele não está. Eu acho que ele não está configurado para conseguir passar o slide, porque se eu apertar aqui, ele deveria passar, e não está passando. Né?
Enquanto eles estão arrumando as coisas lá, vou dar uma corte para vocês, tá?
Passou. Agora foi.
Agora foi.
Os alunos da pós-graduação que se inscreveram
Fica testando.
pós-graduando para fazer disso.
Acho que demora um pouquinho.
Dois cursos em seguida. Precisam assinar uma lista de presença, ok? E vocês, mesmo assim, quem é pós-graduando aqui? Se inscreveu como pós-graduando. Ok. Tem alguns nomes faltando. Então, os que estão faltando, tem seu nome ali, tá bom? Depois eu vejo com a Daniela o que está faltando aqui. Eu escrevo seus nomes. Para vocês terem o certificado, não o certificado, os créditos das disciplinas de pós, ok? As disciplinas de pós é temas especiais em genética. Então, genérico inclui os dois tipos. Mesmo assim, quem é da pós pode, que na verdade deveria, na minha opinião, assinar a lista de presença que conta com um certificado de extensão do curso de capacitação. Aí vocês têm as duas coisas, né? O certificado de extensão e os créditos na pós. Tá? A extensão é para todo mundo que está aqui, independentemente. Mas existe uma palavra-chave, a Valéria pode explicar um pouquinho melhor depois para vocês, que vocês precisam incluir na hora de receber a presença, tá bom? Uma espécie de Google Forms. Então, mesmo aquele que vão assistir assincronamente, recebe presença, desde que tenham conhecimento da palavra-chave, que é dada no final da gravação. Você tem esses títulos, né? Tecnicamente, claro que você pode tentar enganar, mas não seria útil. Um pouco sobre mim para quem não é desse programa. Quem não é do programa de pós-graduação em genética? Levanta a mão aqui. Ok?
Vale. Você podia falar na frente da câmera, então, e já se direcionar para quem está online também, por favor.
Ok. Então, vou me apresentar para vocês que não me conhecem, talvez. Eu sou a professora Angélica, eu estou aqui no programa de pós-graduação em genética, responsável pela disciplina de epigenética e também trabalho na área de genética molecular humana. A minha colega, inclusive, e amiga, com a gente reparte a sala, nós duas somos responsáveis pelo laboratório de genética molecular humana, juntamente agora comigo com o professor Leonardo e com a professora Karen Brown, que é da patologia, e a professora Daniele ficou responsável pelo curso de genética forense e eu pelo de genômica populacional. Ok? Então, eu queria convidar vocês todos a fazerem perguntas quando vocês quiserem. A gente tem tutores para ajudar vocês com qualquer dúvida. Então, uma das tutoras está aqui, queria que você se apresentasse. Vem aqui na frente, tá bom? Professor primeiro também. Espero que esteja me vendo.
Boa tarde, pessoal. Meu nome é Luciana, eu sou bolsista extensionista e vou ficar como tutora aqui do curso, então estou às ordens.
Isso. E a Ludi, ela entende bem o inglês, então, se quiserem fazer perguntas em português, que necessitem de tradução, ela pode ajudar e eu posso ajudar também. Tudo bem? Então, é isso, né? Além da Ludi, tem outras pessoas que estão sendo tutoras, mas de forma mais assíncrona. Mas estão aí também, deixa eu passar o contato de todo mundo para você. Esqueci alguma coisa que seja importante mencionar?
Só a chamada, depois eu posso explicar no meio, acho que provavelmente no meio do intervalo das duas aulas, você disponibiliza a chamada, mas o que é bem importante deixar claro é que cada aluno é responsável por assinar a chamada. Se der algum erro, né, e não for registrado a presença de algum aluno, a gente não se responsabiliza por isso, né? Então, verifiquem na hora de assinar a chamada. Vocês devem receber um e-mail de confirmação, né? O registro que vocês submeterem, vocês vão receber no e-mail de vocês. Caso alguém não receba, avise na hora. A gente não vai contabilizar nenhuma chamada depois da finalização do curso.
Ficou claro? E para quem é da pós-graduação e que está nos assistindo, se você fizer os 2 cursos, você tem direito a crédito na disciplina. Tecnicamente, a gente está recebendo as assinaturas de forma presencial. Se você quiser, de outra maneira, teria que conversar com o colegiado aqui do programa de pós, ok? Para receber os créditos, como é que a gente faria.
Eu acho que é importante, mesmo quem está assinando presencial, são os alunos da pós daqui, também preencham a chamada online que a gente vai estar disponibilizando.
Isso mesmo. Ok? Preenche sempre chamada online, sempre, para que eles ganhem certificado de extensão. Ok? Queria apresentar, então, para vocês, já está gravando, né?
Já está gravando, pode confirmar, Salve, por favor.
Então, todos os que estão, principalmente online, e mesmo os que estão aqui, precisam ter assinado o termo de uso de imagem e voz, ok? Por quê? Porque a gente vai manter essas aulas disponíveis. Inclusive, o professor francês que disse que vai nos permitir usar o PDF, né, de slides, etc., por até 2 semanas. Então, caso apareça o seu rosto, a sua voz, etc., você precisa ter dado a tua anuência de que está tudo bem, ok? Senão, pode sair da sala. Ok, eu quero apresentar para vocês, então, o professor Franceschi Calafel, poderia vir aqui, para que eles possam te ver também. O professor Franceschi, até ontem, eu conheci apenas online. E eu tive o prazer de conhecer a esposa dele, a professora Helena Bosch-Fuste, na Alemanha, em um simpósio da Keystone sobre biologia evolutiva humana. Né? E ali, realmente, depois da minha apresentação sobre o projeto que eu conduzo na minha própria população, se estabeleceu um vínculo, uma amizade, vamos colaborar. E o programa de internacionalização da CAPES, isso é importante frisar, né, que infelizmente está acabando, acho que você é o último, nos permite trazer professores de fora. E quando vi que havia muitas vagas ainda para pesquisadores visitantes, eu falei assim, eu já sei quem vou convidar. Né? E mesmo só me conhecendo online, o professor francês disse que se aceitaria de bom grado. Então, para nós é um grande privilégio ter um dos maiores nomes na área de genética de populações e genética forense na nossa universidade. Ele entende tudo em português, mas isso é verdade. O índice de citação do professor francês é 70. Né? Isso porque, gente, não é só o número de artigos que você publica, é a qualidade desses artigos que conta. Né? O quantas vezes ele é referenciado por outras pessoas. Artigos de qualidade. Em uma época, né, onde a gente trabalha contra os falsos moinhos produtores de artigo, que falta muita qualidade, muitas vezes, na ciência, é realmente algo que demanda muito investimento de trabalho duro, ter um índice de citação assim. Né? Então, para nós é uma grande honra e um estímulo
That only means that I'm old enough to have him producing.
It isn't only that he is too old. In reality, he's very young and he has many jokes too. And I learned with him a lot. So I hope you will enjoy. Agora a gente vai continuar em inglês, mas ele entende muito bem, como eu falei, o português, não precisa traduzir nada, né? E o espanhol, se vocês quiserem, vocês podem usar tranquilamente espanhol e portunhol para fazer perguntas. Ok? Tudo bem? Quem quiser fazer as perguntas no chat, depois a gente vai checar todas as perguntas aí também. Ok. That's it.
So, muito obrigado, Angélica, for the presentation and for the invitation to be here. It's a number of first times. The first time for me, first time in south of the equator, first time in South America, first time south of the Tropic of Capricorn, and first time in Brazil. So and yeah, I'm really delighted to be here. Just because we enjoy a lot always telling what we know about. It's not fun at all knowing or gathering knowledge if you cannot disseminate it. So yeah, I really enjoy teaching what I know, what little I know, and it's and this visit also comes in the frame of a scientific collaboration with Professor Angélica. And let's hope that this is the beginning of a really fruitful relationship. So, and of course, desculpe, eu não falo português, and here's the Duolingo mascot angry at me because I've been practicing only for a month. So I can say things like prazer em conhecer a você, and o gato bebeu leite, but not that. But since I because different circumstances in life, I happen to know four Romance languages. The fifth one, it's not that difficult for me. If you speak really slowly in scientific terms, I think I understand Portuguese, and so I hope this is not a barrier. And please interrupt me, ask questions if something is not clear. Please tell me. If what I'm explaining is too basic, it's something, we know that, please let me know as well, and we will go fast. And so about the formal structure of this course, it's divided in nine aulas, and if roughly they are about 1 hour each. Some of them will be shorter, others will be longer. And I think that we can take a 10-15 minute break between each one, and then we'll be done. Okay, so a little bit more about me. I work as my affiliation is that I work as at the Institute of Evolutionary Biology, which is a joint venture between Universidad Pompeu Fabra and CSIC, which is the Spanish Academy of Science. And I'm also a professor. I'm primarily actually a professor at Universidad Pompeu Fabra in Barcelona. This is the biomedical research park. This is not an optical effect. We are really almost on top of the beach. From my office, which is about behind here, I can see this is a hotel, and I can see the tourists in the hotel using the swimming pool, and I'm working, and then and sometimes I go back home in the subway, and the subway smells of sun lotion, and if I go to work too early on a Monday morning, there's still people coming up from parties. Anyway, so let me brag for a little bit. This is probably in terms of scientific production, is probably the largest the largest scientific institution largest biomedical institution in Southern Europe, and we are blessed by the location. We shouldn't have the right to be there. It should be a hotel, but they say but it helps us in recruiting. Once we want to recruit someone, some important name and data, when they ask about their paycheck, we say, "Oh, look at the beach." And so this is not that important where I work. It's more important what I do. And since it's been quite, in a sense, it's been quite monotonous, my scientific career. I started my PhD in 1991, working on human population genetics at a time where DNA didn't exist, and we had to resort to blood groups and to HLA antigens and whatnot to characterize human genetic diversity. Later on, I kept working on the same with new tools that could address new questions. And I also took an interest, particularly in the more methodological and statistical aspects of an applied branch of population genetics, that is forensic genetics. In a way, it's forensic genetics. You can imagine that it's population genetics sometimes with a population of size 1, the individual you want to identify. And so let's start with this with the first course, human population genomics, and in 2 weeks we will continue with forensic genetics. And in human population genomics, the general idea that what I want to convey, I don't know if learn is even too ambitious, but what I want to convey is mostly about the basis and the methods, both more biochemical, you'll see in the first class now, and particularly statistical methods that we can use to address some different questions. We will learn how we can approach population genetics to learn about the history of a population or set of populations, about their structure, about gene flow, about how their population size has changed. And we'll see also applications of that to, of course, in a separate course to forensic genetics, but we'll touch slightly on aspects such as parts of that may be of clinical relevance in a few steps removed. And sometimes we want to learn where a particular mutation that causes the disease originated, or we want to, if we have an isolated population, what can we know in general about the genetic risks for their health, and how this is related, how this has the risk of being turned into a stigma. Oh yeah, you come from that redneck isolated population, you are sort of red creepy. That's why you have these genetic diseases. And well, we'll try to fight that and to justify why they have not more, but quite often different diseases. Something that I repeat again, and again. And their diseases stand out not because they are more, just because they are different. And a quick example: the Roma, so-called gypsies, have their own diseases, but they don't get cystic fibrosis, which is the most prevalent genetic disease in European populations, so on and so forth. We'll get to it.
Can I ask you something?
Yeah.
The Roma are the state that we call here in Brazil ciganos.
Yes.
Isn't it?
Yes. It's apparently, well, Roma is sort of a consensus name that has emerged in the scientific community because, particularly in English, some Roma resent being called gypsies because it's a name that is associated with some stereotypes that you can imagine that they are not positive. But in other cases, for instance, in Spain, Gitanos, they are proud of their name. Don't call them Roma; they are Gitanos. But still, it's names are a sensitive matter. So and particularly coming with and we have many scenes to atone for in this field, in which we had a sad tradition of working with populations, not even with populations, working on populations from a distance. You go there, take the samples, do whatever you like, never come back, never take into account their interests. So this is something that we should try not to do. And the first thing that we should agree with those communities is how should we call them?
Yeah, and I want to tell you about one thing that happened with me during my PhD. So I finished my whole PhD, and it was the last day where I was defending. But it was really a small group. It was only so with my professor, another professor, and it was about populational genetics. And I was talking about the athlete. And then the guy from the human genetics branch told me, why have you used this name? I said, that's wrong. Yes, it means people that eat raw flesh. And I was like, oh my God.
Yeah.
What is correct? The right name is Inuit.
Yeah, it's something quite typical that these are called exonyms, the names that other communities give you, and sometimes they tend to be quite derogatory, because the other ones because we are always the good ones. We are always better than the others, so we tend to call others with bad names. So. And this is in general what I will try to achieve. So you'll see that I will go relatively heavily on methods, but rather than just explain one single or a few projects, I will give a general idea on methods. Quite often, then I will give examples, and the last class will be just examples of different projects, of different papers in which the methods that I have used, have told you about have been applied. So we will start now with how we get these variations. This is something that I work mostly. I work completely on my computer. This means that the results, the basic genetic information comes already to me. And I think that I should know, and you should know, how this was produced in the first place. Because this will inform about what you can do with that, what does it mean, what type of technical biases might be, what's every type of technique most appropriate for, and so on and so forth. So we'll see about different types of genomic information and how we manage to gather that information. What type of techniques are used to retrieve that? I think that's important. Then we'll touch on the very special two very special parts of the human genome, mammalian genome in general. Mitochondrial and the Y chromosome, they have their particular properties that quite of intersect nicely with some of the questions we might have related to sex. Here's another name. I will be talking about sex, about the X and the Y chromosomes, about the biological determinants of sex, which are something different from gender, which is how one constructs personally and socially like this. And they should not be confused. We shouldn't impose sex on gender, and we shouldn't impose either gender and sex, which is happening right now, and that, in my opinion, it's not adequate. You cannot ignore the fact that some people have two X chromosomes and other people have an X and a Y. You start to see expressions like, well, I won't go there. It's not important. So you get the idea that why this is relevant. And we'll talk about haplogroups and filogeography, and we'll see a couple of examples of this specific, actually kind of social information that this provides. We will actually go from what is strictly sex, the X and the Y's and the eggs and the sperm, to something that's really social, like when a man and a woman marry, goes to live with whom, and that's a social role. So anyway, we'll touch on that later today. Then comes three really classical lessons in every population course: the forces that shape evolution. How variation is produced by mutation, and it's channeled by recombination. And I think again that this is important. For instance, mutation gives us the molecular clock, gives us a clock in which we can date events, and we can measure then the genomic information with history. For tomorrow, we will see all the main forces of evolution that have to do with demography, that's population size and population movement, and then we'll touch on natural selection and what it means and what it doesn't. We will talk about positive selection, which then, of course, refers to adaptation to different diseases or environmental circumstances, but also, of course, there's purifying negative selection. What happens if to the variants that are detrimental to the people who carry them? And then comes a lesson that hope I managed to teach it right, because it's that kind of thing that it seems hard. We will see some equations. Sorry about that, a number of them. But it's the type of thing that, if you really get to understand that, it's like a revelation. It's wow. I can understand so many different things when I know about when I think in terms of coalescence, or when I think actually backwards from the present to the past. On the CESTA, we will talk I have this sort of statistician soul, and we will keep two different things separate, two different ways of using statistics. Descriptive statistics, which is not the same and is often confused with tests, with actual hypothesis testing. For instance, some of you may be familiar with this, but we will, of course, on Friday, we will touch on that. Principal component analysis is not a statistical test. It doesn't, it describes the data. It cannot be used directly to answer questions. So we will make this difference. And the basic difference is here there's a p-value, here there's no p-value. And of course, many people in science need to understand what's a statistical test and what does the p-value mean and what it doesn't. And we will then we will finish with some case studies on the genomics of human populations. And little spoiler, a good chunk of it will be about Brazil. So, which of course I think you might be interested in, and I'm well, slightly uncomfortable about me not being Brazilian, teaching Brazilians about Brazil. But anyway, it comes. You'll see that what I was teaching was made in Brazil by Brazilian researchers.
Yes.
Okay. So first, I titled this population genomics rather than population genetics. Well, because it's just fancier. Anything ending in omics, it's fancier now, and it's more and it's easier then to get money either from the government or from the general public if you do nutrigenomics or stuff like that. And if that's something you can sell to people. No, seriously now, I will try and focus not on single genes, not on single variants, but on genomes. But on genomes, or at least at the very minimum, here in the mitochondrial ionic chromosome, these two genome regions that have special properties. We come. I was telling Leonardo first that we come from a field in which 20-25 years ago, one gene, 5 SNPs, that was a PhD dissertation. Now PhD dissertation is something like 100 genomes. So we will try and have this general view of how can we use genomes or proxies for full genomes to learn on these fields.
And tell people there something that are online. Oi, pessoal. Aqueles que não são alunos do UFR e não têm acesso ao chat quando precisa fazer perguntas, pode mandar sua pergunta por e-mail, ok? Aí a gente solicita aqui as pessoas que estão na tutoria para ler a sua pergunta, para fazer a pergunta ao professor Francesc. Tudo bem? Pode escrever em português. É isso.
Okay, so right now, just okay. 30 minutes, not really saying anything. So let's start with something. So I will try now for the following 30, 35, 40 minutes. Maybe better 30 than 40. We will see types that how we can access this information in the genome, both in terms of the types of variation we have and of the biochemical methods we can use. I will not go that deep into that. I would give you just a general idea, basically because I'm not going to make a fool of myself in a field that's not really mine. So, but still, these are some things that we should know. So, what are sleep arrays, exomes, genomes? The technologies we use in sleep array typing, exome capture, genome sequencing, and we will start at the very beginning with what came before the next generation. So sample sequencing, and I think it's important that you know about that, and I think in the way you should know about VHS, video types, and some ancient technologies that really allowed us to be here. And that, oh yeah, and that's this and this I will go really fast here because because actually this will be developed then in the following classes. A SNP, or as I learned just last week, that in Brazil you pronounce a snippy, and that's I have also another corner of my soul who's a linguist, and oh yeah, that's an epenthetic E. That's the technical linguistic word for it, when you unite a vowel to complete a word. In Italian it would be a, so in Brazil it's E. And then I understood, oh yeah, that's why USP is pronounced Ushpi. That came there. Yeah, the two neurons I have in my head connected with one another. Okay, SNPs. And simple enough. You know the analogy, the metaphor of the genome as a string of letters with sort of fixed positions. You know where position 3,179,315 in chromosome 3 is, and that could be a G or a C, or an A or a T. Any possible pair of different bases, of different nucleotides, and this here could be the two chromosomes of one individual, or this could be a reference genome, haploid reference genome. We'll talk later on about can we do better than a haploid genome against one chromosome of one individual, and we would, and if that's the case, we would call this the reference allele versus the alternate allele, not to be confused with ancestral and derived. They're not the same thing. Ancestral is what came from the ancestors, and of course it's more likely than not that it ended up as the reference genome. But of course the reference genome also contains derived alleles. And this is something that it took me a while to understand. It is particularly relevant then, for instance, in the Y chromosome, in which it's quite clear that the reference Y chromosome, for instance, or the reference mitochondrial are in a teeny tiny branch and are quite distant from the ancestral mitochondrial or the ancestral Y chromosome. So we have reference and alternate allele. And we can go to a SNP database, and they will tell you that each SNP has this arrest identifier. This is just sort of a serial number. It's the order in which they were discovered. Basic information of what it is in relation to which reference genome. And do we have any sort of prediction of a functional information for it? In this case, it's an upstream variant, 2 kb upstream from this gene here. And we know that because there have been 2 publications on it. So we can go to deep SNP, and of course my favorite part would be that one, the frequency of the G allele in different databases, or here the difference of the reference allele and the alternate allele in different populations with their sources, and we can play with this. Oh yeah, it would be so boring if every SNP had all the same frequency in the same populations. How many SNPs are there in the genome? Depends. It depends because the more genomes we type, the more SNPs we discover. And since we have this European bias that many European genomes were sequenced first, we'll see later on in the example, for instance, about Brazil, that if you go to new populations, you discover new SNPs. So this is an evolving figure. And at the end, it will be more important to rather to have the full number, to have a side frequency spectrum. We'll touch on that tomorrow, probably, or on Friday. That is, how many SNPs exist in humans so that the alternate frequency is between 0 and 1%. How many between 1 and 5? How many between 5 and 10? Because at the lower end, every one of you has their own private mutations. Every one of you contains new mutations that do not exist in anyone else, that were created in the oogenesis and the spermatogenesis that then became new. And there are about, I think, figure is about 20 of those on average here. I think that's the ballpark figure. So unless we sequence everyone, we will know the total number of SNPs. Oh, of course, that's a maximum figure. Every single position in the human genome can contain a SNP. Is that true? No, it isn't. Because in some positions, if you have a SNP, you're dead. Mm-hmm. So in some positions are so crucial for life that we will never see them as polymorphic. Because if you cannot breathe, you are not alive. If you cannot produce this organ or the other organ, you're not alive. And we tend to, well, that's let me be more drastic. You weren't even born in the first place. I was thinking, oh, I was about to say, oh, because we only sequence living people. No, that's wrong. We sequence ancient DNA as well, and we will touch on that as well. Anyway, so the point here is there are many. And we can pick and choose, and we can then think of them in terms of their frequency. And so in the 1,000 genome projects, 84% of that would be 5 in 6 of all SNPs have a minor allele frequency below 1%. So most of them are rare. And only about 10% have a minor allele frequency above 5%. So many of them are not that polymorphic. We have a debate with clinical geneticists about what's a mutation, what's a polymorphism. Do we have to count here just frequency of also effects? Don't worry about names right now. So it's important also to understand about functionality of a SNP, and some things are trivial, like we know about of course the genetic code. We know which we know the exons of the genes and whether a particular SNP in the coding part of a gene will result, will result in an amino acid change when that gene is translated, or whether it's synonymous, it will result in the same amino acid. And then, of course, there are intronic genes, there are intergenic genes, and here we have a harder time characterizing function. We can predict splicing sites, we can predict, of course, promoters and so on and so forth, but it's not as easy as it is with studying genes that go apply the genetic code. So here we have an example of this SNP, which is a change of an A for a G at chromosome 4, position 42,001,654, related to genome reference CH38. That results in the substitution of a methionine by a valine in the N-terminus of the zinc T9 transporter. So that's referred to in when you translate that into protein to the MAT50 val substitution. And it has the A allele, which is the methionine allele, is in blue in these pie charts, and the G allele is in yellow in these pie charts, and it's looking interesting. It has a clear geographical pattern. You see, the A allele is much more frequent in African populations than anywhere else. The G allele is much more frequent in East Asia than anywhere else, and then in particular it goes also to the Americas, and Europe is sort of an intermediate position. And this does something, and this does something to zinc transports. So this is the beginning. Just may quote slightly how the story continues. This has this has actually been adaptive, and it has to be, and it sort of interacts with your dietary zinc intake. So
depending on the genetic variant you have, you will need more zinc in your diet.
Yes, you will be. Yes, let's put it the other way around. You will be able to survive better, and if the local content of zinc in the soil is lower.
Okay.
Because we will take zinc from mostly from plants. Plants take zinc from the soil, and the zinc content in soils is different in different geographical regions. But still, this is a paper by Elena's group, and you'll see more than that. I will see later on the only textbook that I will dearly recommend. That if you are interested in this is something that's really good, that touches much better than I will do most of the contents that I will present, which is a textbook by Jonathan Richards. It's free. It's legally free in the internet. You see in a couple of hours the reference. For the rest, if you are interested in particular stories, in particular that I will just touch on, you will always have the references. Most of them are also free, are open access. If some of them are, and you have difficulty accessing them, let me know, and I can send them to you. I had on the first slide my email address. For anything now or in the future that you think that I might be of assistance, please email me.
Thank you.
Yeah, and So precisely these days, I might not be that quick, replying. I've been kept busy, still doing wonderful things, but usually this is not a promise, but 24 hours can be a reasonable response time. Anyway, so the message here is there's additional information that 99.9% of it is not mandatory. But if something attracts your attention, you will have the references to read the papers or whatnot.
May I ask you something
Yes.
about the slide?
Yes.
We see that in Africa we have much more adenine. Okay, so
Yes.
methionine is the position, particular positions changes to almost the opposite in
Yes.
Europe and elsewhere, and it almost disappeared in Asia, and then it reappeared. The adenine again in North America. There are some populations where it gets the overhand again. Is
Yeah.
this related to genetic drift or to natural selection?
Neither.
Neither.
Because these are not native populations. These are the populations in the 1,000 Genomes Project.
Okay.
So, for instance, these that here are African American.
African.
These that here are people sampled in Barbados.
Okay.
So their blue alleles come from straight from Africa, and the yellow here comes either from Europe or from the ancestors of Native Americans.
Okay.
So it's
So these are the migratory routes in.
yes.
I think it's genetic drift also.
Yeah, of course, it can be in some cases, and something important. This is really exceptional.
Okay.
SNPs in the human genome don't look like that. No, they don't. And since they don't, this is why we know that this is a strong candidate for regional natural selection. Your average vanilla SNP has maybe it's, I don't know, 55% in Africa, 50 in Europe, then 45% in Asia. It doesn't change this that much. But when we quantify difference, this comes from this comes from top. This comes from the top 1 per 1,000 of the FST of variation in the human genome. Yeah, this is an important message. SNPs, SNP allele frequencies don't look like this usually. Okay. So, there are methods that would allow us, and that traditionally allowed us to genotype SNPs one by one. We might have been interested in one particular SNPs. There are you could amplify them, sequence about 300 bases, or there were other methods that will allow you to multiplex that. Now genotyping millions of SNPs, it's easy, and well, depending on the situation, it's not that expensive. We are talking about $100 per sample that will get you 1 million SNPs. Bit more. There are companies who make money by, and we will touch on that in some clusters, it will appear in the frontier genetics part. There are companies that sell to the public their genotypes for about 1 million SNPs for about $100, and they make a profit of that. Yeah, maybe a little bit more than that, make it 150. But it's in that ballpark. And we can do that, and we can cover the whole genome, or at least nice parts of the nicer parts of the genome, because there are some difficult areas, and we don't care about those anyway. And we can do massive genotypings in arrays that contain from 100,000 to 2.5 million SNPs. In these arrays, every dot would be a SNP, and they shine in different colors depending on the genotype. Those are SNPs. The exome is the sum of the exons, the coding parts of all the genes in the genome. Of those that we know about, those that we can know about, design probes for and capture. And it's meant to contain maybe not all the coding variation in a genome, but most of the coding variation in a genome. Again, coding variation is something that we can interpret quite easily in terms of changing one amino acid for another amino acid in a protein sequence. And genome is the entire sequence. Again, entire, we should qualify that there are telomeres and there are centromeres that are hugely repetitive, really difficult to sequence. We don't care about anyway, because they are important. If you don't have the telomere, you get older. If you don't have a centromere, you don't have a chromosome. But those are really basic functions that do not change that much between individuals. So it's all right. So they contain the exons, the introns, the regulatory regions, the intergenic regions, and sometimes we might be interested in things that don't do anything. And again, I'll keep this. Why is important that genome region doesn't do anything? I'll keep that here, and I will take it back when we talk about more interesting genetics. And beyond SNPs, in a certain way, exomes, but particularly whole genomes, afford us the possibility of working with structural variation, which is also important, particularly in the case of variation of clinical interest. Not only clinical, but particularly clinical when compared to a reference, we will always have a reference, and we may miss a piece of the reference in a particular region of a particular genome. We call that a deletion. We have applications that are in tandem, applications that are interspersed in the same chromosome or in other chromosomes. When they go to other chromosomes, they are quite often mediated by mobile elements. Whole kind of worms, it's a whole part of the genomics that I won't touch about in this course. We have inversions, which are structural variation, but it's not copy number variation, because we have the exact same information, just in the opposite direction. But they are important because they tend to inhibit recombination, and then everything that you have within an inversion is sort of transmitted together. And this is also interesting. And we have also the translocation of a sequence from one part of the genome to another part of the genome. And most of those, particularly if they land inside another gene, or if they lose their regulatory elements, they will tend to cause disease. Now back to back to genotyping. SNP arrays. In population genomics, we quite like the genome-wide human origins array because it was specifically designed for population genomics. And what does it have that we like in population genomics? But they use a number of individuals from different populations across the world to as sort of as a discovery set. The human origins panel contains SNPs that are present in one or more of these individuals from all over the world. This is a way of avoiding the fact that many SNPs were discovered first in Europeans. So, this avoids the so-called ascertainment bias. So that's nice.
May I ask you something?
Yeah.
It's curious. Why are the French? Why do they have, more than 100,000 polymorphisms? Similar to a bit lower than the Sand Bushmen. Because African people, they are known as high divers, but French, think un-European. Why do they have so much?
No, it's not that they have those. It's how many of the SNPs in this panel came from that particular sample.
Okay.
So I'm imagining this, that they first took this sequence, found these 111,000 SNPs, then came the Chinese, who had 78,000 SNPs that were not already in the French.
Okay.
I'm not sure, but I imagine that this is the case. And then when you come to the Sun in Africa, they have this many SNPs that at that point were not discovered in the previous ones.
Okay.
It might be that. So the general idea is that.
But that's relatively low. I was expecting.
Yeah. It's not how many SNPs each individual is. It's how many SNPs of each individual were used for the path. Because and for a SNP array, we need an array containing immobilized, allele-specific oligonucleotide probes, complementary sequence that will catch your SNP, fragmented nucleic acid sequences of the target of what you want to genotype, labeled with fluorescent dyes. That hey, I'm here. I will shine in this color, and a detection system that records and interprets this hybridization signal. How do we read that the red or the green allele have actually bound to the probe? Sorry. And there are two commercial systems by Matrix and Illumina that may be based on binding, or in the case of Illumina, they use single base extension that releases the green or red fluorescent light. So these are technical. Again, these are technical details, but it's important that to note that our information comes from some place, and they are immobilized in these arrays, in these really small chips, and you can have hundreds of thousands of millions
Obrigada, Lud.
So, for now, SNP arrays are still cheaper than whole genomes. They are easier to process. You get like you get your genotypes, and in the case of whole genomes, the analysis is more complicated, but it focuses on predetermined variation. Often variation was discovered first in Europeans, and we may miss what's not there. What's there? We, in SNP genotyping, we only genotype those SNPs. If an individual or a population has different SNPs, we won't see them. And that's the usual justification in theory, in our paper factory, that you first go for SNP arrays. Then at the end of the paper say, oh, if I had money for whole genomes, I could do this and this and that. Then ask for money to do this and this and that, and then do this and this and that. And once you have whole genomes. So, a bit of ancient history now. We started at the beginning with sample sequencing, and in which we can copy DNA sequences in the lab with primers, nucleotides, and DNA polymerase PCR. We can separate DNA fragments with electrophoresis. We know about size, about electrical charge. How DNA is for acid, which means has a negative charge, and it goes to a positively charged electrical pole. And Frederick Sanger discovered that DNA synthesis can be blocked with labeled dideoxy nucleotides. That if you have a dideoxy nucleotide, it will incorporate to a sequence in a way that's complementary to the corresponding base. So this with A's and A's with this and C's and G's and C's and C's. But it will stop DNA synthesis. And putting all this together, here's the idea. I can incorporate new nucleotides, but when I get to the one that's complementary to the dideoxy nucleotide, it will stop, and it will not grow any longer. So we do this. We have 4 lanes, one in which we have amplified with ACGT and labeled dideoxy A, ACGT and labeled dideoxy C, ACGT and labeled dideoxy G, and ACGT labeled dideoxy thymine. So let's imagine that our sequence is this one, ACTGATGCC. Actually, what we will obtain is the reverse complementary, starting from here. The complementary we will obtain T, G, I, C, and so on. This is the complementary one. So, what will if I have these 4 mixes? Here we will incorporate a T and go on. Here we will incorporate a T and go on. Here we will incorporate a T and go on. Here we will incorporate a T and we will stop. Part of the amplified sequences will stop. They will encounter this labeled T here. So we will find a bond here at position 1 in the T lane. So it proceeds, but since it also has an unlabeled T, it will proceed in all the other lanes as well. And now the next to stop will be here in this G, which is complementary to this C here. And next it will stop here at the A lane, position 3, because this A will be complementary to this T here, and so on and so forth. In the original formulations in the 1980s, labeling was not fluorescent, it was radioactive. So it looked like that. This is like this is the G channel, the C channel, the T lane, the A lane. So the sequence, if we read it from left to right, would be C, G, A, T, G, T, and so on and so forth. Now we jump to the late 1990s, and this was all automated. And I introduced you my colleague Raquel Casal, who works at the Genomics Core facility of RTPF, in front of her automated sequencer, which does this next thing in which, like rather than electrophoresis, we have capillary migration, and at the end there's a fluorescence, this fluorescence detector, and the machine has 96 different capillaries like that. So we can run 96 different sequences at a time. So 2024, before that, and this is how the sequences look right now, and we do have already written for us what's the sequence. In forensic genetics, we would be interested in the height of each peak, but that's a story for me more later. And we can produce in a few hours 96 sequences of a length between 400 and 900 base pairs. This is sensitive to if there's something wrong with my original DNA, we will see that it looks like rubbish compared to what it should look like. And this is particularly sensitive to insertions and deletions, because if we have here, we have this, what's happening here probably, we have this individual might be heterozygous for an insertion of a C here. So we have two sequences that one of them is longer than the other one, but they are running in parallel and it's a mess. But we see that visually. So what are we using Sanger sequencing in 2024? It's still the golden standard for sequencing quality. If Sanger sequencing says the sequence is that, the sequence is that. And if another method sees a different sequence, the other method is wrong. If you're working with Sanger sequencing for 40 years now, and it's the gold standard for quality. We may not be interested in whole genomes, but in particular mutations of polymorphisms, then still Sanger sequencing is a cheaper option. And it's also the gold standard for fragment size determination. If we are interested not in knowing the sequence, but how long our fragment is, then capillary electrophoresis is quite useful. And this is how most of the genotyping of STRs which are used in forensic genetics is still done. What came next is starting about late 2005, 2006. We got the next generation, but since there's the next generation, we should probably properly call this massive parallel sequencing.
Let's say something. The standard sequencing is still gold standard.
Yes,
Still.
yes.
What are the reasons do you think?
Yeah, because there's a trade-off in the newer methods for quantity over quality. So if you can sequence something many times, probably you can afford then to have more mistakes, because because sometimes you will get it right if you do it many times.
Billions. Okay.
So remember my analogy? I don't know, probably most of you are too young to know about this. In the 1990s, there was VHS and Betamax and different standards for video types, and then the winner was VHS. In the late 2005, 2006, there were different technologies for massive parallel sequencing. There was 454, there was ABI, and the winner was Illumina. That's the technology that I wouldn't say that it worked best, but it, at least in the end, it dominated the market. Yeah. So we have gone from 96 sequences of 500 base pairs each to 50 million of 300 base pairs each. That's a huge jump in quantity. First, we start by sonicating our library, sonicating the DNA we want to work with. Sonicate means breaking, shattering with glass, as a soprano might break a glass with her voice. Yeah, to the design fragment length. We just chop it to pieces, and we, this is a basic recipe. I won't go step by step, but you need to add different types of sequencings at the end. Some of them have to do with technical reasons. Others will, for instance, the indexes will allow them multiplexing to combine, for instance, sequence DNA from different individuals. And since you label them, then you can know what fragment came from which individual. And it's all done in these flow cells with this fancy bridge amplification method. I won't go into the detail. There are more interesting things than that. And then there's the actual sequencing, and then it looks like this, it's like a children's game that you have, you know, that you can put these things on top of each other and then have your drawings. If you're interested, here's a commercial video on YouTube in which Illumina, the company, explains graphically how this works. And what you get is not a sequence. It's not the sequence. It's a file with millions of 250 base pair sequences. That's called a FASTQ file. Then you align that to a reference genome, and then it gets you the bound file. This is a particular bound file in which see better here. Each gray strip is one of these 250 sequences that are sort of stacked on top of one another. This is the consensus sequence for all of these. And when compared to the reference sequence, this program is highlighting the differences with respect to the reference sequence. And important concept in massive parallel sequencing, the average number of times in which a base pair is sequenced, that's called the coverage. On average, how many gray stripes are stacked here? That's called coverage. and it's important to have large coverage. And here is an exceptional case. Here I cheated here. I used mitochondrial DNA. And in mitochondrial DNA, sometimes you can have coverages of the order of 1,000 to 2,000. They,
Not nuclear DNA.
not in nuclear DNA. In nuclear DNA, what companies will sell you is 30x coverage, in which every position in the genome has to be seen on average 30 times. Let's say, why 30? Shouldn't 2 be enough? You can see one allele on the other allele, no, but this is random. And you may get all the sequences from the same allele. Even 30x may not be sufficient, because the probability that if you see is that, imagine a heterozygote, you have 2 different alleles. What's the probability that all 30 sequences come from the same allele? 1/2 times 1/2 times 1/2 times 1/2, 30 times. 100 and that would be 1/2 to the 30th, which is 9.3 times 10 to the minus 10. That's the probability that if you sequence 30 times one position, you will only see one of the alleles. But a human genome may carry 3 million heterozygotes, 3 million lactose. And you want to get all of them right. Not just one. This is the calculation for one site. What about for 3 million sites? Then the probability of making at least one mistake is this. This would be the probability of being wrong once. And sorry. This is probability of being correct once. This is the probability of not catching, of being wrong once. So 1 minus this is the probability of being correct once. But you want to be correct 3 million times. This up to here is the probability of being correct 3 million times. If I do the complementary, then this is the probability of being wrong, of not catching a heterozygote, of one of the 3 million heterozygotes, of not catching at least one of those. And this comes to almost 0.3%. This would be the upper error rate for 30x sequencing. And of course, there's a way of making this figure smaller, which is increasing the coverage, but that's more expensive. So this is sort of a compromise.
Okay.
This is the compromise. Do I want to spend my money to increase my quality? Am I okay? Am I comfortable with this figure? Or would I rather spend my money in sequencing more individuals? This is where the compromise comes.
Because if you see an allele for the first time in one genome, but if you sequence many individuals, you hope to see at least in one of these individuals this allele again, which will appear for the first time in one of your genomes.
Yeah.
So it's a way of being more sure that it's not an error. It does not appear so many times.
Yeah, and this is the number of times you would be wrong. Probably wrong in 3 out of each 1,000. But it works the other way around. Maybe the most likely would happen most likely as that you would see as a reference from a zygote, an individual who carries who's heterozygote. And that's not, and it's quite unlikely that you would see really new alleles. just because of that.
Okay.
The errors in new alleles come from other way and from other sources of error rather than coverage.
Okay. But this is the coverage of Illumina. And platforms you may expect different figures.
Yeah, not exactly.
Not?
Not exactly, because if you go to companies like BGI or Microgene in Korea, they will tell you, we will run this many flow cells. If you give us more individuals, your coverage will be that much. If you give us fewer individuals, your coverage will increase. But in any case, the price is fixed for by flow cell, and it will cost you the same. And you have to decide. But yeah, they will give you this figure. If you want 30x, you can fit these many individuals.
Okay, so it's established.
It's
Quite.
for exomes, it's higher than that. For exomes, it's about 100. X. Yeah, because exomes are much shorter. And that's good also in genetics when you want to be really certain of each factor.
Yes, about the pathogenic variant that appears in exomes.
Yes.
Okay.
So if that's your interest, then exomes are also a good compromise. If you are really focused on pathogenic variants, that's a market niche for genomes, for exomes. So, and we'll have the BCF file in which we will be told out of these 30 times how many times a variant has been seen and how many times the other variant has been seen. So there's a lot of bioinformatics going on there. And that's, you need many more bioinformatics skills to deal with full genomes that you need to deal with your STRs or whatnot. And we tend to recruit PhD students among those who have a master's in bioinformatics. At least they know how to work this, that we have tend to teach them the wonder of discovering new things in population genetics, but they know how to deal with full genomes. So, Illumina sequencing is used for whole genomes, for full exomes, and for metagenomics as well. Metagenomics in general means sequencing everything. Everything, not human DNA, but let's put in the machine a sample and see what turns out. A sample, like for instance, a sample of feces, and you would get via and you can identify by comparing two references: human DNA, food DNA, and DNA that comes from the gut microbiota. Then you can match that also to genomes of those bacteria, and you can identify who is who, how many species you have, the diversity, the particular species that you have, and so on and so forth. You can do that, for instance, also with skin samples, with samples of any part of the human body. You can do that with, and now they are starting doing environmental DNA, particularly in ancient DNA contexts. In exome sequencing, we have to, we have this added step of capturing first the, there are commercial probes for exomes, and we capture those, and then we sequence. Now, we may want to move on from a reference sequence to something that's more complete. That a reference that contains also in itself variation, both geographical population variation and structural variations. And we now aspire to have T2T chromosomes. T2T means from telomere to telomere and anything in between. It's often said that the sequence of the human genome, well, of course, it's true that the sequence of the human genome was published in 2001. But that was not the human genome in two different senses, because it was a sequence of someone or of bits and pieces of different individuals, but every fragment of it came from just one person. And also in the sense that it was an approximation, it was not finished. There were still some pieces that didn't connect to one another, particularly around centromeres or at the end in telomeres. So now we want to have it all. We want to have T2T chromosomes. This is particularly going in the Y chromosome, which is particularly difficult and convoluted with lots of structural variation. And now there's this sort of new idea to represent genomes not as straight lines, but as graphs, as something a little bit more complex that we have to wrap our minds around, because we need to incorporate three things: individual variation, structural variation, and T2T chromosomes. And we're going to do that with long-range technologies, which is the main, and well, we'll see different applications, but now the next generation is long-range sequencing. We want to go from sequencing every the genome in bits and pieces of 200, 300 base pairs. We want to go up, and we'll see in a minute that it's feasible. We want to go to 10 kilobases at a time. And we'll see the technologies and why this is relevant, why this is important. Long-range sequencing is much better for assembly and mapping, particularly in repetitive regions. It's much easier to solve a jigsaw puzzle if you have larger pieces than shorter pieces. Much easier to overlap. But it's more expensive and the error rates are higher. Though we'll have some, you'll see, fancy applications. There are two main technologies, PacBio and nanopore. PacBio is single molecule real-time sequencing, and nanopore means having the DNA go through a nanopore and nano-sized pore. And both technologies look out of the fantasy of a science fiction writer. Okay. Now I will read this, and I don't know if I would understand it any better than you will, because PacBio is based on the zero-mode waveguides VMW in a nanophotonic confined structure that consists of a circular hole and aluminum cladding film deposited on a clear silica substrate. The ZMW holes are 70 nanometers in diameter and 100 nanometers in depth. So ZMWs allow observing nucleotide incorporation in real time for a single DNA molecule in each ZMW hole. So it's basically, it goes like this. The single molecule is DNA is immobilized in each MW. These molecules may be about 10 kb long, and you get your results going through this CMW. It's up to 100,000 base pairs with high priority rates, but the application of population genomics can be genomic sequencing from scratch in non-reference species. We will talk mainly or almost exclusively about humans, which we've had for 23 years of reference genome. So there are many interesting species for which we don't have a reference genome, and it's much easier to do that with if you have long fragments. It's much easier to work with copy number variants if you have long sequences. And you can do haplotype phasing in vitro rather than in silico. And for instance, this is something that one often wants to do in clinical genetics. You want to know the context of a particular mutation, what's around it. This will tell me whether two patients have two independent mutations, the same mutation but that appeared more than once with different origins, or the origin is the same. And we can use also the length of the shared haplotype to date that mutation. So with short read sequencing, if this is a mutation of interest, we will go maybe one step up and down, but not more than it. If I have long read sequencing, then we will reach much further, and we will know that now this mutation goes with the C and the T, and since the T will go with the C and the A, we will say that this mutation goes with the A and C and the T. We can solve the puzzle of the haplotype phasing. Now, works by monitoring changes to an electrical current as the line passes through, or it's also a different technology for long fragment sequencing. Yes.
To go back only for the TechBio, so there are fragments, but they are longer.
Yes. Yes.
Okay, I thought that TechBio was really the whole molecule, but from tip to tip, really.
No, it's
long fragments.
It's long fragments. It's almost 100 kb, but it's not like
okay.
What can be the whole molecule? That's actually nanopore.
No. Okay.
Yeah.
Thank you.
Because it's the whole molecule that goes
Through.
It's same function. And so the sequence is spread continuously, and you have a continuous output. So the machine is telling you in real time A, C, D, G, D, G, D, 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 and you can wait until the end of a particular chromosome. And some nanopore devices, like the Minion, are this big, are small and portable, so they can be used for educational applications, to go in the field, to be used in labs in remote areas where it's more difficult to get the whole contraptions. So I think I'll leave it here. Yes.
As well.
It's something that's that again is a trade-off. It has some technical difficulties, but the advantages of having long fragments sort of compensate for that. It's a matter of it's a commercial matter. Maybe you could have a new generation of nanopore or PyPio with in which their engineers have managed to reduce the error rate, but that would come at a price. That would be certainly more expensive. Or I don't really know. It might be the case that that these errors are not above the feature, that they are intrinsically, that they cannot be addressed. But I suspect that they can be addressed, but at the price. And the price will probably then put them out of the market. So they are basically telling their customers, yeah, acknowledge that making mistakes. Look, this is what we are giving. This is what we are giving you.
One time for each base, right? Because the object pass to the core and you just one time and you have coverage.
É verdade. Eu vou dizer que a Marina responde que ele é bem abaulado. Se fosse feito, é muito mais rápido. Ele é bem mais difícil.
Extensão.
Estava no mudo. Eu acho que não é aqui. Aqui tá tudo. Tá sem som.
So it's fascinating. In reality, the long range sequencing helps us to identify big deletions and insertions
Yes.
that we cannot see with the normal massive parallel sequencing of the working again.
So what comes next is, well, it's again more of the same. Some examples that we used in the set. So I would leave in place this first. I would finish it quite light. So I would finish this part here. Any more questions? Yeah.
My first question is about the use of the microarray. So you showed us what is the problem to use the microarray, that so the SNPs there are from, many of them are from European population. But do you think that it's still worth it to work to perform microarrays?
It depends on the problem.
If you don't have the opportunity to perform,
Yeah, if you're interested in demography, in population structure, in even yeah, they are still worth it. And for that, I would recommend human origins, and that because they overcome this issue with European ascertainment bias. But if you really need, and some methods require a complete description of variation, then you have to pay for or not full genomes.
You mentioned that the standard now, like to sequence by Lumina, is the coverage is 30 times. And what are you using to perform in your experiments or?
Yeah. Again, in our line of work, we use whole genomes when we need complete descriptions, and there are some methods that require that. And we may also want complete descriptions when we want to see all of the functional variation. And for that, we will only require exomes. But sometimes, since you may have one gene here and one gene there, you need to know what's happening in between. For instance, in branch of homozygosity, you really need to see to sequence everything.
How many times?
In, oh yeah, 30x is fine for our instance. And for exomes, if we are interested particularly in detecting pathogenic, then the common coverage that you are sold is higher than that. It's about a 100x. More questions?
One more. You presented for us many techniques to sequence the genomes. Which technique are you using right now?
And I've still used SNP arrays. In some cases, we are using whole genomes. If we can afford them, we use whole genomes. There are some techniques that really require them. But we have also worked recently with exomes as well. So, or for instance, in a particular case, we were just interested in the Y chromosome. Sometimes we can use microtubule line or the Y chromosome as byproducts of whole sequencing. It's, quite often these are they are in the garbage bin of the genomes, because particularly the Y chromosome is not interesting. It's not interesting at all. It's really few exceptions in medical applications. So sometimes we have said, oh, can we have please your Y chromosomes for this fantastic biobank paper you published with 500 human genomes, and that's interesting. But on a couple of occasions we have captured the Y chromosome and then sequenced the Y chromosome only, and that allow us with the same money to pay for more sequences. So that's something that's again. In the job of being a scientist, this is quite often what you do. You manage a budget, and you have to make do with what you have. More questions?
Well, my name is Marco, and I'm sorry if it's a question that you will explain later. Here with Beatriz, we work with analysis of some sequencing data, and we arrived at the point to analyze the population structure. And we discussed which databases are more recommended for us working with Brazilian populations. And we first used the 1,000 Genomes project. And then some researchers here recommended the HGDP, the Human Diversity Genome Project. But researching, I found the Genoma. I don't know how. Yes. And there are a lot more samples, like 100,000 more samples in Genoma. So I don't know.
Yeah, there are different uses for that. If I recall correctly, there was a paper recently in which they sort of unified with the same standards 1,000 genomes and HGDP. So for population genetics, that's perfect. Genomas, that's or genome A, whatever. The numbers are astonishing, but the population coverage is not. Yeah, and most of them come from medical genetics, and sometimes their way of describing populations is a little peculiar. So, and or the demographic metadata can be kind of poor. Or the Genoma, you have seen that they put in the same label, I don't know, South and Finland. Well, it's not the same. Finland is a homogeneous population, South Asia is 100,000 different populations or castes or lineage belonging to 5 linguistic phyla. So Genomas is good if your question is, this variant that I see in Brazil, has someone ever seen it before? Has it been described and where? That's perfect. If you want to, if the question that you're asking your exams is, for instance, this piece, this genome fragment, where does it come from? For that, I would use the combination of 1,000 genomes and MTDNA.
That's my question. Thank you.
You're welcome.
Do you know the Abraham?
Yes,
Yes.
we're using it. She told us.
Okay. This is a Brazilian database. But there is one problem, Abraham. You know which problem?
The bias, the phenotype bias.
Yeah, phenotype bias. The elderly, healthy people in São Paulo.
Yes, they're mostly whites.
Yeah, that's true. And they are healthy and they are all already.
Yeah, I think that was, I don't know.
So maybe
First time in Brazil. So, but I would imagine that old and healthy would skew white in São Paulo, even wealth inequalities.
Tell me what probably.
Yeah.
Maybe Japanese.
They also have many more patients than the rest of us.
Yeah, it's true, in São Paulo. Another question? It's like another question online. Will we have a coffee break or will we?
Yes.
Yes?
Yes. I was about to. We've been, for your sake as much as for mine, we've been going on for an hour and 58 minutes. It's time for a break. I suggest 15 minutes, back at least, that's okay? Back at 4:05.
Okay, 4:05 here.
I'll be here.
Sim. Tem que falar aqui, né? Aqui tá a câmera. Aqui, né? Não, o L. Aqui mesmo? Ok, pessoal, então a gente vai dar um tempo agora de 15 minutos. 4 horas e 5 minutos nós pretendemos continuar, ok? São 4h49 agora. Não, 3h49 agora, então 4 horas e 5 minutos a gente continua aqui. E no final a gente vai entregar a lista de presença para... Deixa eu ver. Tem aparelho de genética aqui, tá? Não precisa. Você é uma aqui, ó. E qualquer coisa vai curtir. Pode voltar também. Fala com gente. Tem que mandar esse da cápsula agora.
Ser rápido e efetivo. Então, tu pode estar na evolução. Eles fazem variados sobre a
Não é só.
Eu não. Ah, você vai dizer. Ai, então, vou. Tudo bem. Pode falar contigo. É só mais câncer.
Obrigado por isso. Claro. Eliminar.
Eu estou mas não tem que trabalhar comigo. Chegou a me atropelar, então, e não deixa. É isso. Quando não deixa, tudo que a gente sabe assim, tem mandando uma resposta para cá, você deixa que a carta falou assim, o projeto não pode seguir adiante porque a UPS não está na lista das universidades do grupo contribuidor. Eu estava na lista do dropout da CAPES. Agora eu estou falando de recurso, lotar esse conhecimento que eu não posso ser coordenadora. Nenhum desses projetos multinacionais, porque eu sou professora de pesquisa. Que a questão conversa. Eu não falei literatura, mas a gente tem que dizer, escuta, que incoerência é essa? A pessoa que professora de pesquisa, isso não significa que ela está o trabalho dela é diferente. Eu realmente assim, daí, assim, tipo, eles não sabem tudo. Se eu tivesse uma vez alguém no governo, eu ia dizer, gente, agora são pessoas que minaram caras. Deixa você ver o que é. Vem aqui, gente, quer explicar como são as editais de você? Assim, não tem mais coerência. Então, agora eu estou terminando aqui, espero que.
Sim. Olha, essa experiência, eu poder entender mais ou menos
Como funciona, né?
Deus que você não quer saber. Eu colocava uma pessoa de salvar. Tinha horas que ele não salvava. Ele apagava tudo que eu tinha feito. Eu falei, coloca uma coisa e salva.
Deixa eu olhar. É difícil, você pode pensar o dia inteiro, eu tinha que fazer. Isso tipo assim, assim, dignidade eterna, com a Genômica. Porque se ela na sala de estar dela, não é. E eu na minha sala de estar. Se eu estivesse paralelamente, mandando uma protocolo, mandando a outra, uma dos fluxos e outra. Mas nunca.
É um sistema muito bom. Muito.
Mas a minha ainda por cima, o quê? Cadê o você não viu o edital do
I'm working on maybe I'm not even there. So
But at least, I think,
yeah, some interest of being counted that there might have been.
yeah,
Okay. Nice figure 66. That's the year.
it's very,
That's the year I was born.
yeah. I'm going to make some coffee for myself, and I'm coming back soon.
Oi, pessoal. Só para conferir, Eduardo, vocês estão vendo a apresentação em tela cheia?
Sim.
A Lud está aqui. Desculpa.
Sim. Obrigada. Ó, agora está funcionando o passador.
Certinho. Então, estamos gravando, já estamos no nosso segundo grupo de slides para a segunda parte da aula. Daqui a pouquinho o professor Francisco vai voltar. Eu só vou diminuir o volume.
And how was your experience this morning?
Can you yeah, of course you can imagine that. I could imagine that we went to this restaurant in Moretus that they had sort of an outside patio, and there were a number of titi monkeys that were actually waiting for us to feed them a banana.
Alright.
Oh, and the views and the landscape. Yeah, it's like, well, yeah, really. Now I am in Brazil.
Yeah.
Because, cities, yeah, this is treats you as differently, and you won't city and totally different and so on. But the landscape, the community say that. Maybe I wasn't quite expressive in expressing my admiration because like
And you see much more.
You have luck because it's not common to see the monkeys there.
That's really something as well.
Sorry?
Can I connect to some Wi-Fi network?
Yes.
Oh, it should be. I should be
If
connected.
Ron is working?
Oh, yeah, it is. Now it is. Yeah, it is.
Okay, if
So,
not,
yeah. So my phone company sent me the fees for the roaming charges for Brazilian. Oh, okay.
Yes, but you need Wi-Fi.
Yeah.
And you, it's 4:05, and people are not back. Look at this picture, they're really wonderful. Essa chave aqui. Tem uma muito parecida dessa, obrigada por me ajudar.
Mas eu quero que você faça.
Introduce you. Come on. Sorry, I forgot to introduce my different. My colleague, but you said it's also sorry for I told my mom. For the genetic laboratory, because working the HIV context, and also the whole transplantation program. Part of it, defense. Just a little more.
I have very little experience in that field because 10 years ago we used HLA antigens to as population markers, but they are quite tricky because there's collection going on and some groupings can be a little weird and it's so easy to over-interpret. But still, it's
Comfortable with the population and we have a lot of data. We see it we start to explore these things.
Yeah.
Trying to look for general population. But it's very
so lately, and it's not that for some particular tests with the actually written is particular bias. But then you tend to, for instance, when you try and see these, when people not want to find it, we see that we can find the different answers to this. And actually tends to bias towards Africa.
Really?
Yeah, just by itself, it's not that the usual interpretation would be about having actually teachings of African origin would be it's not just that it's a more diverse population.
It's the right here trying to look at those we're trying to look at those regions of the city and we were over the don't have to use the and in the North region you have more the explanation so you're trying to look at and also to look to the children population and so there's the other machines that are not influence are and sorry that everybody out there when they're students that you already they're trying to secret change so the number of
Yes.
Yeah. We have to look at the capital class. And so much we have illness or most interesting. Yes, in one of the so you have here.
Let's start again.
Yeah.
Oi, pessoal, nós vamos começar agora. Tudo bem, gente? Vamos continuar aqui.
Again, please, if I'm sort of speaking over your heads and there's something that I'm saying that really prevents you from following, interrupt and all that, and which is that. And so, yeah. So we will resume now with, I guess that we can pick up the pace now. Otherwise, otherwise we will get too late and jet lag will kick in. I don't want to.
So we don't want to go so late today.
because I don't want to to think of what time is now is right now home. I'm more of a morning person. So without further ado, two very special regions of the genome that function as markers for themselves. Mitochondrial DNA and the Y chromosome. I will go, this is sort of the index for what's coming next. I will touch on where does mitochondrial DNA come, its structures, in content, NUTs, which are the nuclear copies of mitochondrial DNA, some reference sequence, why, its role in sex determination, its evolutionary history, and so in both cases, we'll talk about concept of haplogroup, which is just mean groups of haplotypes with usually with a particular geographical distribution, and what they mean and what they don't mean. We'll talk about how mitochondrial DNA and the Y chromosome are informative for sex-specific processes, and we will illustrate that with a case of the Americas, and how we can use mitochondrial DNA and the Y chromosome to trace genealogies, and what can we do with tracing genealogies. So we've been told again and again and again, or at least at the beginning, that you get half of your genome from mouse and the other half from dad. Well, that's not exactly true, as you probably know. Setting now aside, we'll touch on that when we move to forensic genetics. Setting now aside the X chromosome, we know that men get their Y chromosome from their fathers, and both men and women get their mitochondrial DNA from their mothers. They do not recombine. I have to qualify that. The Y chromosome does recombine in its tips and the so-called several regions. It does recombine with the X, but for most of its length, it doesn't recombine with anyone. And in the case of mitochondrial DNA, there are examples in other species of recombination. There are some researchers who think they have found recombination in human mitochondrial DNA, but they haven't convinced many people. So, this lack of recombination means that mitochondrial DNA, except for mutation, travels down genealogy through the maternal side, and except for mutation, the Y chromosome also travels in a piece down the paternal line. Two important features. It's in mitochondrial DNA we have thousands of copies per cell, and that justifies its applications in ancient DNA and in forensic genetics. There's base by base, nucleotide by nucleotide, there's more mitochondrial DNA than nuclear DNA in the genome. Not altogether, but sort of the internal coverage of each base in mitochondrial DNA is on the order of thousands, whereas for the rest of the genome is 2. For the Y chromosome, it's actually 1. For Y chromosome in whole genome studies, you get half the coverage, because although there might be exceptions, there are men with 2 or more Y chromosomes, but usually we have 1.
And this makes a huge difference, practical difference I never thought about.
Yeah.
Right.
Yeah. So, I was a grad student when mitochondrial DNA started picking up, and we were joking among ourselves how the first slide in any presentation about mitochondrial DNA was a circular molecule of mitochondrial DNA. So we ended up calling the donut. So here's the donut. Yeah. And mitochondrial DNA, mitochondria have their own DNA because they are former bacteria. There are some features in mitochondrial DNA that make it close to bacterial DNA. And first of all, the fact that it's circular, like bacterial genomes. So, and of course, it was Lee Margulis who proposed symbiotic theory for the evolution of organelles and how many of them, and particularly mitochondria and chloroplasts, are former bacteria that sort of lived in that state living inside eukaryotic cells. Mitochondrial DNA is a really compact genome. It has we can list quite easily these genes that makes about seven genes coding for enzymes, 2 for ribosomal RNAs, 22 tRNAs in white, these short sequences. It's so compact that some genes even overlap, like here. And there's only the origin of replication in here, and in the so-called control region, which, as we'll see in a minute, contains hypervariable regions. That's 1, 2, and 3. So actually, this would be position 1, and it will grow like this. 2 up here, we will next to position 1. We have position 16,569. That's the length of the reference sequence of human mitochondrial DNA. In many cases, your sequences are quite likely to be 1, 2, even 3 base pairs long, because the reference, the mitochondrial DNA, is actually 1 base pair shorter than average. So again, references are not dogmas, are not written in stone. They are just someone's sequence.
I have a question regarding the mitogenome. Do you have an idea of which parts or which genes are more nuclear ones and which were more bacterial origin?
If I remember correctly, and let's, if I remember correctly, all of them are of bacterial origin. And in fact,
Nuclear origin.
No. All of these genes are bacterial in origin, and actually, many of the original bacterial genes migrated to the nuclear genome.
Okay.
There are.
I thought that there were also some nuclear genes in the mitochondrial DNA.
No, it's actually the other way around.
Okay.
Because there are many genes in the nuclear genome that control, that express, sorry, that code for proteins and enzymes that are used only by mitochondria. Yes, as I was about to say, so many proteins that are exclusively in the mitochondria are encoded by the nuclear genome in these different functions. And there are there are fragments of the current mitochondrial DNA that from time to time migrate to the nuclear genome. They are not lost. They are copies of what remains still in the mitochondrial genome. So we have about 33 parallel sequences with over 80% sequence similarity and of a greater length than 500 pp. And this is the mechanism. So this sometimes is a technical, it causes a technical issue because they have their own life, they mutate independently, and if you're not careful, you will mistake them for the actual mitochondrial genome. And and you see sometimes some bizarre sequences in databases
And they can have the range of an entire mitochondrial sequence. The entire mitochondrial sequence in size.
Yes. Yes. It's this is not this in an carcinoma, but that's not fixed. That's not fixated. It's something that happens on occasion. It's not that everyone has a copy of the mitochondrial DNA in there. There are some that they are fixed, but they are relatively short. They are about 500 people.
How do you think that this happened?
I don't know. Ask the surveillance.
A's that travel to the genome or transcribed and then inserted. I'm traveling now in imagination.
Yeah. And this is the history of mitochondrial DNA, of the human knowledge of mitochondrial DNA. First human sequence was published by Anderson in 91. It's referred to it as an Anderson sequence. The current reference sequence is the same one, just with a couple of mistakes corrected. And there was something of a ghost nucleotide and position 3167 that actually didn't exist. One like sequence, it's 18 years later, it wasn't there. So now the reference sequence has a gap. So we can align it with the original sequence and preserve the same numbering. People who are experts on mitochondrial DNA will recite to you, this haplogroup is made of positions this and this and this and that. You have this, so we do that. So we wouldn't like to change our reference frame just by one base pair. So they and that's something that sometimes causes technical mistakes if you are not aware of that, or the programs used to align mitochondrial DNA are not aware of that sometimes. Then came there in 2012 that using we know the Neanderthal mitochondrial DNA sequences, and those were used to root the human mitochondrial DNA tree. And we know what now we know what is the ancestral state at each position of mitochondrial DNA, and this is the RSRS, the reconstructed sapiens reference sequence. These are references, but but on top of that, we want variation. And the first variants were described by RFLPs in 1987. We're talking here studies that were done before PCR. And this means that they needed, I think, almost a kilo of fresh material. What part of the human body, some of you already know, please don't spoil it. But what part of the human genome weighing about 1 kilo they use from people, not causing any damage. Placentas. Yeah. They ask for donations of placentas, and they could extract the blood from the eye that they needed before, because this was for PCR. And this, people don't realize now how important PCR is. That you can, that or otherwise that you modern genetics would be impossible without PCR. And with sequencing and PCR, getting ahead of myself. And sequencing about 600 base pairs. This was confirmed, and it was confirmed that the sequences that split first in the mitochondrial tree were found only in Africa, and of course in people of African descent elsewhere. So, mitochondrial DNA is more variable than nuclear DNA. They, given its nature and the fact that it's not covered in histones, makes it more accessible to mutagens. And at the beginning, when sequencing was really expensive, it's mitochondrial DNA which the genome portion that gave the most information for the same amount of sequence. And within the mitochondrial genome, this is nucleotide diversity. This is how variable DNA is. This is computed in 125 pp windows. So along the sequence, we have here hypervariable regions 2 and 3, and this is hypervariable region 1. Funny thing, this is outside any so-called hypervariable region, because this is just one base pair. This is 16,519 that mutates by tracing, and it's kept at the frequency of about 50%. It's almost random. We don't even use it in when doing phylogenics. So here I'm zooming in, and here we see from 1624 to 16,383, we have HBR1 from position about 70 to 360. This is HBR2, and this is something relatively new that comes mostly from forensic genetics. In the 400 and 500, we have hypervariable region 3. Now, the current approaches in sequencing is that either you sequence either mitogenome or your sequence roughly from you sequence the entire control region, about 1 kilobase here. And this is also, if you do that, that's quite informative as well. So I expect that if we now sequence the control region of the 25 people that we are in the room, we will get quite, I would say that we would get, just with this kilobase, about 22, 23 different sequences. And if we sequence everything, as we say in Meton, I bet to be that we will find 25 different sequences. I bet to be. So, using RFLPs, this is polymorphisms that affect restriction sites this is an archaic language. I mean, there are particular SNPs, and let me try to translate this into modern terms. There are certain SNPs that will affect how bacterial enzymes can cut DNA. Those are called RFLPs. And for many times, for quite a long time, these were the only way that we had to characterize DNA variation. So, applying this technique to MTDNA in samples of Native American origin, it was done like with this for the first time with Soronian and colleagues, and in '93 observed that they found 4 main patterns of presence of or absence of cutting sites for RFLPs, and they called that Haplogrupos. These haplogroups, when they were associated with CR sequences, with clonal region sequences, and besides that has particular variants in control region sites, and they go together because mitochondrial DNA does not recombine. That's why we have this association. And finally, these were confirmed and refined using full mitogenomes. So now we have this reference tree that you can find here. Now it's been frozen since 2016. There are attempts now. There are now 3 times more sequences available in databases than when this was drawn. So now there are some attempts to refine it, but it's been frozen as a reference for the last 5 years. And these are the main branches. Each branch has a name. And Haplogroups should get, but it got messy, a systematic nomenclature that starts with a letter. I don't know if you can see here. Now imagine that you go to your ophthalmologist. Which letter is this one? This is a J, and there's a branch of J which we call J1, and there's actually another branch down there that's J2, and the first branch of J1 is supposed to be J1A, but J1A no longer exists. Anyway, it's J1B, and then we alternate letters and numbers to designate the sub branches. So we have J1, J1B, J1B1, J1B1A, J1B1A, 1A1, and so forth, and so on and so forth. These are just labels. But those labels are important because once you cross that information with who carries which haplogroups, haplogroups have quite a distinct geographical pattern. There are haplogroups here in bluish colors, these labeled here in blue, that are found in Western Eurasia and North Africa. Others here in this dark green are more typical of North Asia, or this red collection here, hugely diverse haplogroups, are found mostly in sub-Saharan Africa. This is not a mistake, because Madagascar was first people from Indonesia.
It's beautiful.
Yeah.
Biogeography.
Yeah. And here in this, where are you? We have, I guess it's supposed to be this color here is the Americas. Haplogroups are not people. Your haplogroup does not determine where you are from. Where did you get your mitochondrial DNA? Your mom. Who got it her from? Her mother. Go back 10 generations. your genome is a mix of 1,000 of the genomes of 1,024 different people. Because in 10 generations, there's no consanguinity, you will have 124 different ancestors. But you got the mitochondrial DNA just from one of those people. So, I know I'm saying this again and again and again, but this is something really crucial in forensic genetics, in which it's absolutely wrong to infer the geographic origin of an unknown sample just from mitochondrial DNA. But again, this applies at the individual level, at the collective level, at the population level. Of course, we can say things. Here's more of the same. Different haplogroups are found at different frequencies in different world populations. For instance, here's when we take 2,700 Brazilians from something like six, 7 different states and different regions, north, northeast, center, south. You get about 34% of mitochondrial DNAs, mitochondrial DNA sequences, belonging to haplogroups that are being found most frequently in Native American populations. 21% Europeans, 43.7% Africans, 0.4% Asians in Sao Paulo. These are most like the Japanese descendants.
The Brazilians were collected where?
These are the DNA to the Brazil project. I have that. I don't know. They were from different states in a way that looks to me, to my non-Brazilian eye, as a relatively wide net. We will see that on Friday. I will show the slide with the exact origin of this.
Okay.
We can wait a couple of days. Okay. I defer then my answer to that because I don't want to be here. And since mutation rate is known, we can add a temporal dimension. We know when haplogroups diverge from each other, and to these migration routes, we can add them signs. Some of these cliffs here are really old, in the thousands of years. Other cliffs are much more recent. For instance, some of these branches here, for instance, New Zealand was first peopled in 700 CE, just 1,300 years ago. Now, onto the Y chromosome. This sort of thing of a chromosome, we shouldn't forget an idea of how important and how relevant to life the Y chromosome is that half of the population lives happily without one. Thus, so this means that, yeah, there are few genes in the Y chromosome that if they didn't exist, you would be perfectly well. There's just the SRY, which is the switch that triggers the development of the embryo as a male. That will then trigger the cascade, the developmental cascade that will end up with an embryo having male genitalia and the capacity of producing then sperm cells. So this is this is a schematic portion of the schematic representation of the non-recombined portion of the Y chromosome. As I mentioned, part 1 and part 2, the cell autosome of regions 1 and 2, do recombine with the X chromosome. The rest is a block. With lots of heterochromatin, there's actually, I was about to say that it was visible at the naked eye, but well, visible under microscope, there's variation. Well, more variation on the size of a Y chromosome. Now, if you have a big Y chromosome, that doesn't make you, I don't know, more attractive. It doesn't matter at all because it comes from different numbers of repeats of this heterochromatic region. And then there are different types of coding regions here from genes that all came from the X chromosome at different times. The Y chromosome is actually a determinant. Everything is just a determinant of X chromosome. The X chromosome that can be slow and degenerated, but this has happened on occasion, and it's not. And the small chromosome does not mean that it should be a male, because in birds it's the other way around. Remember that WW birds are male and W, what am I going to speak in American or British? W, Z or Z, birds are females.
And do you think this is because of the Miller's bracket hypothesis? Yeah.
Well, I don't know that issue that well, but and then there were then some of these regions are descendants from these original X chromosomes. X chromosome, others came at different times, and we can recognize that in the amount of difference that we have between the X and the Y regions. So the X degenerate is contains 27 genes and many in multiple copies. Some of them can give some phenotypic effect. There's some copy number variants. There are particularly deletions that result that are de novo, that appear first in that individual who carries them, but because the effect on that individual is making him infertile. And we have SNPs and we have STRs on the Y chromosome. And for quite some time, most of the information we have on the Y chromosome, sorry, came from STRs and short-term repeats, because they are much easier to time than SNPs on the Y chromosome. They have these repeat complex structures. Actually, in the next class, we'll touch a little bit more on STRs. And they are even duplicated, so these are still complex repeats, but they can be quite variable in size. That's what we isolated in. There are about 80,000 SNPs in the Y chromosome, and this can be used as well to construct a phylogenetic tree with sort of the same structure, the same nomenclature than in the case of mitochondrial DNA. This combination of a letter number, but this can sometimes be so long that an acceptable alternate denomination for these haplogroups is to refer to one of the terminal SNPs. So, and we can there are no standard panels type SNPs in the Y chromosome. In every paper, you will see different sets typed with different methods. That was an area now we have moved on, and now we can either capture about 9 megabases of the so-called callable sequence. Callable means that, yeah, you can get sequences from those. From the rest of the chromosome, such a mass of duplication and structural variant that's it's pretty much uncovered.
Impossible to analyze.
Hmm?
Impossible to analyze. Actually,
Yeah. Yeah, there are some papers that have come out lately on that, but they use PacBio. But they use really long fragment sequencing. The rest of the models, we either capture this or use the leftovers from whole genome sequencing. It's hard to work with. You can have the coverage, but still the information is there. And funny thing, now there's an official scientific why haplogroup tree. This is much more complete, much more there's much better precision here, but this is the the ISOGG Y chromosome tree. And the funny thing about it is that it was compiled by citizen scientists. It was compiled by people who started going to 1,000 genomes, looking at the Y chromosomes, constructing tree, and there were amateurs who did that first, rather than people who are like me, who are paid to be a scientist. And they have been maintaining this web page for the lot of knowledge. No one is paying them to do that. And it's a pity sometimes that this work is dismissed in scientific circles that, oh no, we cannot use that because it does not come from peer-reviewed science. Information is there, and actually this is the official tree. You may recognize the same sort of the same graphical style as in the mitochondrial DNA, but this certainly comes. This is sort of a simplified version of the other one of this. May recognize the names of the first two authors in this paper. We were among the first to recognize that that SDR variation on the Y chromosome is deeply structured by haplogroup. That there's much more difference in SDR allele frequencies between haplogroups than between populations. And this is again the work of an amateur who turned that to a Bayesian method in which you can enter here YSDR haplotype, and they will predict. These are the probabilities that it came from one particular haplogroup. And this is so much the case that there are scientists who mistake these predictions for actual haplogroups. And no, the haplogroup is something that's defined with SNPs. And there's some room for confusion. This is, again, this is actually my YSDR, my genotype from some YSDRs, and I know which is my haplogroup for the Y. And here the prediction is 100% and it's 100% correct. But there are no mistakes, particularly for haplogroups that have the small sample size. So we should take that with a grain of salt. And again, Y chromosome haplogroups have a strong geographical structure. Here we have in different colors, different geographical origins, and we see that it's not 100% perfect, but one color for a continent predominates in most of these branches. So we can do the same exercise that we did with mitochondrial DNA sample now is half the size of Brazilians, and we have now the geographical origins of white haplogroups in Brazil, and it's now 70% European, 25% African, 2.5% Latin American. This 2.2 Asian. There might be some ambiguity there in what Asian means. It might include some that come from Western Asia, from the Middle East, for instance. So if we put the two charts together, we start learning something about demography. We feel this is the percentage of ancestry in blue mitochondrial DNA, in orange for the Y chromosome, and you see that it's completely different. The Native American proportion is much higher in the mitochondrial DNA and the Y chromosome. It's the opposite in Europeans. Also more mitochondrial DNA of African origin, more mitochondrial DNA on the Y chromosome. And of course, this is the history of the Americas. Who came here? Who killed whom? Who raped whom?
Done in this book done also collaboration with the group of Sergio Pena? No? Okay.
This is me sort of scavenging on the DNA Brazil project. As I told David, please can I have a look at the mitochondrial DNA in the Ys? And she gave me the data. All I have done in the mitochondrial DNA and the Y chromosome of this sample is shown here.
And it's still going on. And there's lots of illegal metal extraction in the Amazon basin. And now we see natives that are blonde and blue eyes because of this.
Yeah. And that's why you cannot infer where someone comes from or what he or she looks like based solely on mitochondrial DNA or Y chromosome.
Someone from master's group and look at the mitochondrial DNA and why DNA, they're completely different. Most of them white, most from European, and the other mitochondrial DNA, South American, and this, and they saw it's yeah, different often. There's the white foods and also pretty different yields and regions.
Yeah, of course, I did not discover this. There's been tons of papers about this. I just used it as an example that I thought would resonate with this audience. Because if we take the samples from the Americas in the thousand genomes project, I have this Afro-Caribbean from Barbados, who are mostly African, both in mitochondrial DNA and Y. We see also this huge asymmetry between mitochondrial DNA and Y in the general population of Medellin in Colombia. Larger proportion of Native American ancestry in the also on the Y in Mexicans living in Los Angeles. Also larger proportions of Native American ancestry in Lima and Puerto Rico is a mix of everything. So, general populations, urban populations in the Americas are mixes in different proportions of these three origins. So,
can you go back to this last slide and looking and thinking on culture? Maybe this has also something to do with the development of the Native American culture when the Europeans arrived. Because if you look at the Mexican,
yeah. Of course, these are the Incas, and here you have the Mayas, the Aztecs, and whatnot. And whereas in Colombia, probably the native population had a smaller sample size, and in the islands, it vanished quite quickly. We with the Genomas did something like that for Cuba, and it was known for historical records. And then 50 years after it was discovered, the native population had been completely eradicated. But the current population of Cuba still preserves 20% of their mitochondrial DNA sequences are of Native American origins. By Native American cultural diet, they mitochondrial DNA sequence. So there are many more things that you can do with in respect to demography with mitochondrial DNA and the Y. So you can estimate differences in effective population size that have to do, for instance, in if you count. This is something bizarre that you may say it's not something logical. But in many populations, particularly historical or traditional populations, you average the number of children per man per family. No, per man. And the average number of children per woman, they are different. And in particular, the variances are different. There are men who have many children and many men who don't have any children, don't have access to the wealth necessary to be married, for instance. But this asymmetrical distribution may be less asymmetrical for women. And that can be, we can try and see the effects of that on the variation by the bundle behind the white. And we may see, for instance, differences in migration rates. Who migrates either across continents or at short ranges, which would I said at the beginning, in marches in which the couple comes from different places. Where do they go and live? And many cultures, they stay in the group's place. In other cultures, they stay in the bride's place. And these are called, respectively, quasi-local or multi-local societies. And we can try and see the reflection of that in their people. So in particular locality, for instance, you would expect that that seems it's the women who move that would erase differences between populations, and it's certainly the case in Europe. But why that you can tell quite easily. For instance, if you show me the frequencies of white chromosome haplogroups and ask me, Frances, do does this sample come from the Iberian Peninsula? And I would say yes or no. give me the same sample from mitochondrial DNA, I would say, oh, yeah, Spain, or Portugal, or France, or Italy, even the UK, because there's much the difference here in the geographical structure of mitochondrial DNA is much flatter than for Y chromosome in Europe for the case of because of the practice of patriarchality. That is, at short ranges, it's women. But quickly now, so they put these researchers put these to the test in two traditional groups from Sumatra. One of them are local, the other are matrilocal, and funny thing, the patterns of differentiation between the mitochondria and the Y came out the same for both populations. So, so maybe there's a cultural prescription. Oh yeah, you're supposed to do that, but maybe sometimes people do their own thing. So reality may be different from cultural prescription, or maybe it's the quality behaviors are real, or maybe they actually wish to have had a really large impact on these structures. MTDNA and the Y chromosome follow chronological lines. So if we have this analogy, this woman and this woman would have the same mitochondrial DNA except for mutation, and this man and this man would have the same Y chromosome. So we can use that. We can use that in different to answer different questions, to identify historical remains. So in case of Kyrgyzstan, the Tsar Nicholas Romanov and his family, Richard III, Beethoven. In the case of Beethoven, I'm really good friends with a guy who did this, and he had one of the worst times of his life when he goes to do this identification. He had to gather the living Beethovens, and he had to go back to them and tell them the results. And the results were, we have good evidence that this that we have sampled belongs to Beethoven, but your Y chromosome does not match. But they have a hypothesis that is that it was his grandmother's fault. That it was historically known that he was not in good terms with Beethoven's grandmother, that she was an alcoholic, that at one point she was interned in a sanatorium, and maybe it's not the father who was not the father, it was the grandfather who was not the grandfather. So this happens. Oh yes, in the case of Thomas Jefferson, it's like in those papers that title says so. You don't need to read anything else. It's that this is the genealogical conclusion. This was one of the earliest applications. We're talking here that you could do that in the last century. And again, you can do that systematically and estimate extra paternity rates. Let's take a lottery now. Please guess what's the probability that the father is not the father? The average should do for that.
Depends on the population.
Bilingual Europeans.
Average European.
Let me do it easier for you. Let's have a show of hands. Less than 1%. Between 1 and 5%. Between 5 and 10%. Between 10 and 15%. Okay, I need to go faster upward. Between 20 and 30%. Between 30 and 50%. Okay, so I have a hypothesis that it's not confirming this sample, is that we don't tend to give higher probabilities than that. The actual figure is 2%. And we know that in we know that. Sorry about that.
This table isn't so.
Yes, yeah. Yeah. And we know that in two different ways. Oh, here's one way. We know that from comparing surnames and Y chromosomes. We have this is a picture for a general audience. I think that in Brazil it's the other way around, but you can understand that in Spain we have this double surname system. But it's the father's surname first, and the mother's surname the next. So my children with Elena, or her children with me, are called Calafate Bosch. They take the first surname of each one. This means that their children will not be called Bosch any longer. So we have here an extra generation, but in the end, it's many surnames were transmitted. Now the law is much more flexible than that. It's 31 surnames that can be transmitted, but traditionally it was like that. So this means that you can consider surnames as alleles in a locus at the Y chromosome. They are transmitted like that. And you can then analyze them together, surnames on the Y chromosome. And this is a list of surnames that in Catalonia these are the sample sizes that we collect from man with one of these surnames. In many cases with photographic variation. There are also spelling mutations here. You may recognize some familiar surnames here, and or this was a bet that went wrong. I was trying to attract a student to work on this, and I will solve your surname in there. At the end, that wasn't the price wasn't right, and she didn't come anyway. So no, there are good scientific reasons for all of them. This is a selection of different types of surnames. For instance, there are name, and someone one are names of traits. The name means innkeeper, Ferre means smith. There are last names that used to be first names. There are place names. There are Sabatators, shoemaker, and so on and so forth. They are really frequent surnames, not that frequent surnames. So, and these are the SNP types, and so the general idea was we could recognize groups of people having the same geographical origin, the same surname, and the same Y chromosome at a very fine detail. So, if in the same location and the same surname, we have people who did not fit, men who did not fit, we can count that towards non-paternal. Of course, you can think of infidelity, but there's also options, and we have to do some of that. And we have cases in which it was the maternal surname that was transmitted. Quite often, for instance, in single women, the classical sad cases in which a single woman got pregnant and the father disappeared, the baby got her last name. But it also happened at the other end of the social scale. These are different geographical areas within Catalonia and otherwise, and we got different rates. Are people from Girona? No, I don't have Girona there. Are people from around Barcelona or from central Catalonia really more unfaithful than people from Valencia? No, what happened, particularly in the northeast of Catalonia, is that farms and states passed strictly through the paternal line to be, they went, states were not divided, they went to the oldest son. But, and a woman could be married only if there were no male sons. But if a woman inherited, her sons got also her last name, because somehow the name went with the house. So that explains the higher rates there. This is done in Flanders and in Belgium with actual genealogies going back centuries. And here we could match natural genealogy to a mismatch on the Y chromosome, and we could sort of spread this. And there was also tons of information about all the members of the genealogy, such as traits or an indication of wealth. So these are the estimates of non-paternity rates. Here, this is just extra paternity rates because most adoptions were documented and they are not included here. So these are the different estimates in different genealogies, and the rates are higher among the urban low-income people. And this has to do with probably with wealth inequality itself. These were women who were not in a position to say no sometimes. So that would explain this. That's the end of this. Do we need another break, or should we go straight to the finish line?
I think we can go straight to the finish line.
Okay.
But this was amazing. So DNA is not like and there are chromosomes.
Yep.
So it's even mirroring social structure and violence and everything else in a way.
Yeah, there's a colleague of mine in our institute, who's now actually the director of the Museum of Natural History in Barcelona, who wrote a book called Inequality, and it's all about DNA. It's how inequality is written in our genomes. Not as a cause of inequality, but as a consequence of inequality. So now, now we will go back closer to basic biology. We will go back from society to what happens inside the cell. And we go to this basic processes that creates diversity. We can do the previous works because there's diversity in the human genome, because not everyone is the same, and not everyone is exactly the same. Sorry, talking about genomes, not every people. And not everyone's genomes are the same because of two basic forces creating diversity, and these are mutation and recombination. Because in the end, it's mutation that creates diversity. Recombination puts the final touches on it, sort of shuffling the different variants.
Maybe you can try now.
Yes. So we will talk about mutation types. Well, mutation rate estimates, haplotypes, recombination, and linking disequilibrium, and recombination. You'll see that I'll try to pick up the base here, and because some of the parts are not really that important, and some of the parts may be really outdated. So that's why I'll skip them also because it's late. But if any of you is interested in those details, ask me, and I'll give you the sources for those. So here comes the first slide that I listed from this book that I wholeheartedly recommend. A Noveller's Guide to the Human Genome, Introduction to Human Population Genetics, Variation and Disease. And it's a funny thing because Jonathan Pritchard is doing this, and he's really an authority in the field. And he's doing this selfish thing of publishing it for free. But he also publishes every chapter as soon as he considers it finished. So the entire book is not finished. Don't look for the part of disease is still not there. But the part of many things in population genetic variation are there. I would recommend that to start, but also to go far beyond the point where I'm taking you. I would recommend it, especially if everything that I'm telling you is not news. If you want to go deeper, go to this. So it's done, and you can. And it's there, and you can freely and legally download the PDF files. So it's a huge recommendation. And every time you see an illustration that looks like that, it came from that book. That looks like a professor who's really good at drawing, and then, which is certainly not me, then it comes from Pritchard's book. So when DNA is replicated, there might be mistakes, unfaithful copies. We call those mutations. If that happens, they happen in mitosis. These are called somatic mutations. In meiosis, they are germline mutations. They are the only ones that we will consider here because those are the ones that are passed from one generation to the next and may spread in a population. Somatic mutations, of course, are important in their own thing. Like they are cancer, These are SNPs. These are single nucleotide polymorphisms, when you have a mutation that changes one nucleotide for another one. And this type of mutations, single nucleotide mutations, are called transitions or transversions according to their chemical nature. If if we change a purine for a purine or a pyrimidine for a pyrimidine, then we call that a transition. If a purine becomes a pyrimidine or the other way around, we call that a transversion. And that's important because transitions are much more frequent than transversions. In particular, if we have a methylated CpG, if we have a C that's methylated and that's next to G, then there's a chemical process called reamination in which this 5-methyl cytosine becomes a thymine. And then in the it's a C that becomes a T, and that's a transition. These are two pyridines. So then this will have happened, and if we look at the complementary chain, then we would see that apparently an adenine has been turned into a guanine. And there's another type of mutation called replication slippage, in which if we have a sequence that's repeated, sometimes the replication machinery has a hard time matching exactly the two chains. For instance, we have a loop here on the template strand, and we will have here that this copy is shorter than the template. Or sometimes this growing new copy sort of loops on itself, and it becomes longer than it had to be. So this creates and elongates the short tandem repeats. These units that are repeated a polymorphic number of times. They go usually from 1. That's maybe stretching the definition, like to make it 2 to 6 nucleotide motifs, and they are the workhorse of forensic genetics. So we will deal with them in forensic genetics. And this other mechanism, non-allelic homologous recombination, in which we have this green sequence that's similar to that red sequence, and now there's the coupling here is all messed up, and now we start copying here, we start copying here and ending up there, and we may have a deletion or a duplication depending on the relative position of the two chains. Or if we have inverted repeats, they can loop on themselves and create a crossover between them and leads to an inversion. So this is how they how these sorry, this should be a C. This copy number variants and other types of genetic polymorphisms appear. We have deletions, insertions, duplications, translocations, or duplication here, or inversions. Altogether, these are called structural variants. Now, this is how, but how often? Not that often, particularly for changes in nucleotides, changing one nucleotide from the other. And they did that in Iceland, compared trios of father, mother, and son, daughter, and the child. So this is a number of de novo mutations per program. It's higher than the figure that I told you. It's not 20. Each of you has about 60, 65 new mutations. And here's a field in which I would pay extra to have extra quality sequences, because how many of those are actually sequencing mistakes. They would make a huge impact. I think that to check on that, they took a sample of those, and a large sample of those, and replicated them with Sanger sequencing. That's a way in which you can have this golden standard.
That's it. The novel mutations, they were found in probands. Probands for rare diseases was associated with the age of the father.
No, I think it only refers to the child. Child in the sense of descendant, of offspring.
Okay, not related to anything.
No, they did that next.
Okay.
They could then go by type, transitions, transversions, CPC transversions, and they have the rates here. So you see that transitions are an order of magnitude. No, CPC transitions are an order of magnitude, 2 orders of magnitude almost more frequent than non-CPC transitions. Mitochondrial DNA also an order of magnitude higher. Indels not so much. Short-tandem repeats. This is slower than I'm used to think, because they took into account all short-tandem repeats, not the types that are longer and more mutable and that are normally used in forensic genetics. And here's the relation with from the parent of origin, and it's higher for that than for moms, and it grows with age also faster for the fathers and for mothers. So again, this is for SNP mutations. Remember though that for large chromosomal arrangements, the opposite is true. For aneuploidies, for the wrong number of chromosomes, particularly for Down syndrome, you know that the prevalence grows with the mother's age. But again, that may shape our perception, and it comes from the fact that it's from a very unusual type of mutation and aneuploidy that's rarely incompatible with life. There's a question there. How do we know parent of origin? Long reads. Long reads. Long reads actually haplotypes. We looked at the variation that's near it, and if it looks like the variation that the father has came from the father, if it looks from the variation that the mother has, it came from the mother. So here's another example. Is it just fits my ego in my medal? We also did that with Y chromosome STR. Oh, here's the Guzman, who now has a foot in Portugal, and the other foot in Rio de Janeiro. And these are data collected in paternity cases in which they had good evidence to know that the father was the father, but the Y chromosome of the father was slightly different from the Y chromosome of the son due to mutations. And if we take the cases in which there was a mutation, the average age was 37.3. If the non-mutation transmissions, the average age was 31.3. That was a 6-year difference, and there's a p-value. Before that, this is still significant. And I was sort of flabbergasted that in those samples, there were people involved from 18 to to 70 years old. They were men who were fathers at that age, not only fathers, they were already involved in paternity cases.
Another question. What do you think? Depending on the context, on the environmental context of the population you're looking at for the Y chromosome, or for mutation rates, peak mutation rates, would this change the mutation rate? For example, if I work in Bolivia, I know that almost everything is contaminated with heavy metals. Would this change the mutation rate? Until you, was there any work done on this? This would be really something, under the umbrella of One Health.
Yeah. There's definitely tons of evidence for an effect on somatic mutation. Germline mutations seem to be a little sheltered from that. This was done already with survivors of the atomic bombs in Hiroshima and Nagasaki. And if I remember correct, and it's done again in Chernobyl, and and it doesn't seem, of course, it had a huge effect on women who were pregnant at the time. But for women who had been exposed and later had children, I think that the result was that they couldn't see an increase in the mutation. I don't know if I don't know, and I don't know means I'm an ignorant on that. Whether this has been studied, I think it may have, but the problem here, and this is also justified why you can do it more easy in an easier way with radioactivity, because there might be an issue with differences in exposure to mutagens, and you need to have good measures of that, and that's not that easy. Because of course you cannot do that. You cannot do a randomized clinical trial with that in humans.
I think maybe on the melanocytes and looking at the melanocytes yourself, similar genomes.
Yeah, but you need large sample sizes for that to have any, because these events are relatively rare. So you need a large sample size, not of people, but of de novo mutations.
Yes.
And possibly on the same side, there are
It's not easy to do.
yeah, I don't see good an easy good design that will be statistically powerful enough to tease apart those effects. So, question at the back?
I was just wondering, correct me if I'm wrong. I know in Panama we have big research on biometrics. Yes, and cancer. So I was wondering if we have a big animal cell and a big cohort of people exposed and also exposed through life with chemicals, agrochemicals, and I was wondering, I had never thought about.
We had carbonas for me, you had roads down.
Yeah, of course, you need quite high quality genomes. I would start with exomes, because mutations there, of course, I said that all of you have 55, that you have 65, yeah. And yeah, of course, you need pedigrees, and
O microfone foi mutado. Olá?
Okay, but so I was wondering if someone had researched mutation rates because it was very spread in the city, and we all know, like common knowledge in the city, that we have very high Taiwanese rates, and it's different. And we also have many good surgeons to do the separation, but some students, yeah, she had her doctoral thesis on mutation rate, but she used STRs on the Y chromosome, and she didn't find anything, but that sounds very shallow.
Yeah, yeah, I can understand, yeah. You have, yeah, I can understand that, I can understand at least 3 reasons for that. It's easy and cheap to do. Why SDRs like in kids, and why they are relatively easy to and cheap to genotype. You have parental of origin, which comes from the father. And the mutation rate of those SDRs is, the baseline is known, and it's relatively high. So her sample size could be relatively larger because she was bound to see mutations.
Yeah, she is like, and our city has 2 million people.
Yeah. But then the main critique would be what I was mentioning, that in any case, she even if she had found a higher rate, she wouldn't have proven any health effects, because those SDRs don't do anything. They are no Y chromosome. If they are SDRs, no Y chromosome. That's the double definition of doing nothing. Yeah. So and I could imagine a reason for imagine a positive result. The fear that she would have caused the misinterpretation. Yeah. So yeah, it's you see that I'm not liking it, because it's basically just because it's a case in which the methods don't match the relevant question. And it's the basic of good scientific practice. You need to these two elements need to match.
Considering that this was a relatively recent event, it was very concerning.
Yeah.
Because it was like this from going to the river. Some firefighters said, maybe we should throw this in the river. Maybe we should check what's in this plastic bag. And then they discovered it was radioactive. But yeah, from us that we are from the city, we are very concerned about it because the site where it happened, it's still in the center of the city, and it has like this big of concrete on top of it, but we have trees growing on it. Yes, and we have this in the center of the city, the site where it happened, and it's not. It doesn't have any signs, and it's just a lot of no signs. It's a lot of you. How nice is it? And it has like meters of concrete on top of it, but there's no sign like don't
Yeah.
here. And we do have the disposal site 2 kilometers from the city. And sometimes we visit it as kids. But yes, sometimes it feels like people forget it and we don't have much studies about it. I mean, every time I hear something about Chernobyl,
Hey. What's the Netflix show about Goiás?
I think it's being hidden or purposely.
Yeah. I don't know how I know, but I suspect that not many people know about it.
About this accident.
About this accident in Goiás.
Please, can you explain the next slide again? Because it was muted when you went to the next slide.
Yeah. So this was these were Y-chromosome STRs, and coming from paternity cases, and we had the age of the fathers, and we saw that when they mutate, the novel mutation happened. Parents were on average 37 years old. If there was no mutation, fathers were on average 31 years old. So again, this is indirect evidence that that mutation rate increases with the father's age. So, moving on to an important concept. We are talking here about mutation rates. It's not the same thing that substitution rates. And mutation rates tend to be higher than substitution rates, but substitution rates are what we get when we compare to different species, for instance. For instance, we know that the human and chimpanzee lineages split between 5 and 6 million years ago. We compared human and chimpanzee sequence, count the differences, divide by twice this time, get a substitution rate. What's the difference between mutation and substitution? Because not all mutations persist. Some may be slightly detrimental and may be then eliminated from the population. They will persist for a time, but then here will come natural selection with a site and mow down these new mutations, and we won't find them when we compare different species. Oops. And also there are technical issues such as parallel mutations, back mutations. Here there's time enough for a particular SNP to mutate, say, from A to G, and then back from G to A. And somehow we have to account for that. And there are some statistical tricks to do that, but they may not be they may not be do the job completely. Haplotypes. Haplotypes are combinations of polymorphisms that are physically close to one another. Each line here would be one particular chromosome, and the combination that we have of alleles at different sites are what's called a haplotype. Haplotypes are created by mutation, by they are shuffled among them by recombination. So this sort of links naturally now to recombination, and you see here recombination with two whiteboard bands in the nice colors of local Barcelona. And so here's what we have the parental chromosomes, and here are recombinant gametes. And if you don't have recombination, it's quite unlikely, unless the same mutation happened twice in different backgrounds, that you would see more than 2 or 3 different haplotypes in the combination of just 2 loci. So, one property that sort of binds together haplotypes is the Higgs equilibrium. It's a non-random association in a population between alleles at 2 loci. That one allele at one locus tends to go with another allele at another locus, just because recombination has not acted enough to break those linkages. So mutation creates LD, and recombination destroys it.
Good. And there are also different recombination hotspots in the female and the male genome.
Yeah.
Very interesting.
So here's a graphical representation of linkage equilibrium versus linkage equilibrium. You have and here at the sides, the frequencies of the different of the alleles at the B SNP and alleles at the A SNP, and these are the size of these rectangles represents the frequency of the different haplotypes of the different combinations. If it's like that, if it's the perfect product, then we talk about linkage equilibrium. If they are different, here there's linkage equilibrium because you see that big A goes with little b and little a goes with big B. So we may assess linkage equilibrium with some parameter, and we may statistically test. Traditionally, we can take just the difference between the observed and the expected frequency. The observed frequency is what C frequency plus haplotype, and the expected frequency is just the product of the two allele frequencies, like it was here. Here, the frequency of big A, big B is just the product of big A times big B. Here, it is smaller, so difference between this actual frequency and the product between the frequency of big A and big B can be used as a measure of linkage equilibrium. And this is the D is just that, the difference of the observed haplotype frequency minus the expected haplotype frequency. It can be written like that, and it has these interesting properties. Skip. D is D or D prime. When it's 0, it means no LD. When it's -1 or +1, it means completely D, meaning that one of the haplotypes does not exist. But this is quite old news. For 10-15 years, we prefer R or R squared, which is the correlation coefficient between these sort of 4 points. If they are really independent from one another, there's no correlation. Here, this will create this correlation. And this is the equation for R. Again, not that important. R square is more, it has two interesting properties. One of them is that statistically, R square represents the amount of the